my last video, I discussed about the blood group, blood group of four types of blood group. There are four types of blood group: AB, and O. Uh, I have also told in that video that the AB are actually antigen. A blood group means A antigen will be present on the surface of the red blood cells, and B blood group means B antigen will be present on the surface of red blood cells. AB means both antigen. I have shown by one diagram in my previous video. And O means there will be no antigen on the surface of the red blood cells. Means actually A antigen will be, the gene, the allele A, for A antigen allele will produce the enzyme which will actually the H antigen, one single antigen is there, H antigen, which will be ultimately converted into it is the precursor of A and B antigen, which will be ultimately converted into A antigen and B antigen by two different enzymes. I have told that alpha 1,3-N acetyl galactose aminyl transferase, which will convert H antigen into N acetyl galactosamine, and in case of B antigen, H antigen will be converted into D galactose by the enzyme alpha 1,3 galactosyl transferase. So different enzymes are involved for different different allele A and B, and in case of O antigen, there will be O antigen, the individual will not produce the produce enzyme, but this enzyme will not convert this H antigen into A or B antigen. That's why there will be no antigen on the surface of the O blood group, red blood cells of O blood group. This is the common thing, but we are generally seeing that there are four types of blood group are there: AB, but reality is different, because there are more four blood groups are there, because in my previous video, when I am discussing about the occurrence of different blood groups in Indian population, there is a one indication there. I have mentioned that there will be another grouping of human blood is the positive and negative. What is the positive? Positive means O positive, O negative, A positive, A negative, AB positive, AB negative, like that. So there are 8 groups of blood groups are there, and we have seen that the most abundant blood group is O positive, and most rare blood group is AB negative, from the the table drawn in the previous video, shown in the previous video. Now what is the meaning of the we have already known the what is A antigen, B antigen, and O is the O positive, O is the universal donor, and AB blood group is the universal acceptor or recipient. So O blood group can be given to any blood group, and AB blood group can take any blood group. So this is the reason the antigen present on the red blood cells of the particular blood group people. Now the what is the positive and negative? I have told that Carl Landsteiner in 1900 classified the human blood group into four groups: A, B, AB, and O. And after 40 years of that, after 40 years of 1900 means in 1940, this 1940, the Carl Landsteiner and again Carl Landsteiner and another scientist, Weiner, actually discovered this positive RH factor, rhesus factor, actually discovered the rhesus factor in the Indian monkey. The name of the Indian monkey is the Rhesus macassus. Scientific name of the Indian monkey is the Rhesus macassus. They had developed, these two scientists developed, identified the another factor, rhesus factor, which is called RH negative and RH positive. They have discovered in the Indian monkey Rhesus macassus. So this is the thing. Actually, what is the meaning of this RH positive and RH negative? It is also antigen which is present on the surface of the red blood cells again, and this is the RH positive and RH negative indication of that. It is also the antigen antibody factor antigen. It is also antigen. It is called D antigen. Actually, this is called D antigen for RH factor, and it is generally present in the surface of the red blood cells. Which red blood cells contain the this D antigen, which is they are RH positive, and which those people blood cells, red blood cell will not contain on their surface the D antigen. They are classified as RH negative. So positive when D antigen is present, and negative when D antigen is not absent on the surface of the particular group of red blood cells of the particular group. So it is also very important for antigen antibody reaction, humoral immunoresponse, because where RH factor, RH plus, RH positive is there, there no antibody will be produced, because their body will recognize the self antigen from non-self antigen. So RH positive group, in their blood plasma there will be no RH D and antibody or immunoglobulin present in their blood plasma. But in case of RH negative, if any red blood cells contains the RH positive will come during the transfusion of the blood, when any patient need, then there will be problem, because they will be identified as a non-self antigen for the particular RH negative people, and they will start producing the antibody, and they will attack the antigen, the red blood cells, and coagulate them, and clotting can be happen, and it is very risky and dangerous. So this is the simple way, not as critical as AB antigen, but it is the same thing. So we have to be careful during the transfusion of blood, whether we have to be careful that whether it is AB or O. At the same time, we have to be careful that blood group will be A positive or A negative, means rhesus factor is what we have to identify that. And this test, the identification of the blood, so has been done by the particular antibody will be given to the blood for the clotting, and they will be identified like the AB positive, AB negative, O positive, O negative, like that. And then we can transfer the blood, transfuse the blood to the patient. Otherwise, we cannot do it. So it is the simple way of RH positive and RH negative blood. But in the pregnant women, this is very dangerous for the pregnant. This RH factor, RH positive and RH negative. Why we are telling that? When the mother, during the pregnancy, when the mother, it is not necessary that the baby's blood group will be same like mother. It may be different, it may be RH positive, RH negative. But when the mother blood will be RH negative and the baby's blood will be RH positive of any AB, any group, but RH positive, then there it may pose a risk during the pregnancy. So what is the risk? Actual risk, actually, this is the during the pregnancy, this is the development of the fetus, means new baby growing on the there, and this is called placenta. This is called placenta, and this is developed during the pregnancy across the cell wall, across the wall of the uterus. This is a temporary organ where it is developed when the pregnancy is there. So actually, what is the function of this placenta organ? Actually, it is supplying, transporting the nutrition and oxygen to the fetus, means newborn baby, and also takes away the wasted material from the blood of the baby, newborn baby, which is called fetus. Now, what happened if this fetus is RH positive, if the mother blood is RH negative, what happened? This maybe across this placenta, the some red blood cells containing the RH positive factor will cross the placenta and go to the reach the blood plasma of the mother, and mother is RH negative. So mother's body will respond when the red blood cells coming from the fetus to the mother's plasma. They will produce, mother's body will produce the antibody, and this antibody containing plasma, mother's plasma, will go through the again the placenta and reach the fetus blood, and the fetus, they will actually, the fetus blood red blood cell will contain the RH antigen. So this antibody will attack this fetus red blood cells, and this will be the problem. It may cause the severe jaundice, possibly brain damage. So this is the factor, this is the risk factor, and this process is called the antibody will, mother's antibody will clot the baby's antigen. This process is called sensitization. So there is a preventive action is there. Actually, during the pregnancy, the mother will be injected with the anti-RHD immunoglobulin factor by the doctors, so that the to prevent the mother from producing this antibody, which is the antibody, and the action of the impact of this sensitizing effect on the fetus. So this is done, and generally doctors, when the mothers will be only RH negative blood, and some doctors have administered the anti-RHD immunoglobulin injection to the mother at 28th weeks or 34th weeks of the pregnancy, to prevent this sensitizing effect of the on the fetus, and according to WHO, World Health Organization. Actually, this the baby, when our antibody will start growing after our birth. After the birth, the antibody is actually start growing, and it reaching at the peak level at 5 to 10 years of age, and declining after fifth decades. So this is the overall thing. Rh positive, Rh negative is not very concerning, but when we are transfusing any blood, we have to be careful about that, whether it is Rh positive or negative. And Rh positive will be given to Rh positive blood people, and Rh negative blood will be given to Rh negative group. It will be very careful. And during the pregnancy, this type of sensitization effect will actually create the problem. So we have to be careful during the pregnancy, so that the fetus or newborn baby will be not affected adversely. So this is the all about the blood group or human blood group. So I hope that you will like this video. So thank you.